Hi everyone, we're going to talk about the phases of mitosis. Now, you're looking at a picture of the cell cycle. The cell cycle includes a long phase called interphase. GAP1 or G1, S, and GAP2 are all part of interphase. That is the time during a cell's life cycle when it's growing, making new organelles, making new proteins, and preparing extra DNA for the upcoming event, which is mitosis. You'll notice here that mitosis is 4 phases: prophase, metaphase, anaphase, and telophase, sometimes pronounced telophase. After telophase, we enter right back into interphase here. And that, of course, continues all the way around until we approach back to mitosis once again. Mitosis takes up about 10% of the cell's life cycle. The other 90% of the cell's lifespan is spent during interphase. Just want to have a small conversation here about DNA. On the far left, you may recognize the double-stranded helix of DNA. DNA is actually twisted and wound around proteins called histones. Those are shown in picture 2. Those histones help us to organize that DNA into what's called a chromosome. On the far right here in picture 5, it's what we call a double-stranded chromosome or a replicated chromosome. This chromosome shows us that we are ready to undergo mitosis. The reason we know it's ready to undergo mitosis is there are chromatids here on the top and the bottom. There's a total of 2 chromatids here. They are cinched together in the middle. That centromere area there is actually where we're going to connect to that chromosome using mitotic spindle fibers to help separate these sister chromatids in 2. So if I were to draw a line between the identical portions, the left and the right sides of this chromosome are identical to each other. Here you can see again that chromosome, the 2 chromatid arms on top and bottom. Like I said, there's a total of 2 total chromatids here. They are cinched together by a belt in the middle. We call that area the centromere. Now the reason I point that out is because during prophase, you'll notice condensed chromosomes. In the picture on the far left, you can actually see those chromosomes there. In the other pictures, metaphase and anaphase and telophase, you can still see the darkly stained areas known as the chromosomes. You may or may not be able to see individual chromatids, but you can see the dark staining chromosomes. In fact, that's where they get their name from. Chromo means color, some means body. So these are colorful bodies. They pick up stain when we dye these cells. So let's start by talking about interphase. These are actually pictures of plant cell, most likely onion root tip. I know that they're plant cells because these are rather square-shaped cells. Okay, animal cells tend to be more round in shape, at least what we're going to look at. So what I'm looking at is nucleolus. That's the dark spot there, only visible during interphase. And the chromatin, the rest of the stuff in the nucleus, looks like just static. It's not anything that's organized. I can't see distinguished or condensed chromosomes. And this whole structure here is our nucleus. Can't see much else going on in the rest of the cell. Mainly what I'm looking at to identify this as interphase is what's going on in that nucleus. I see a clear and distinct nucleolus, that's that darkened region, and then I see what looks to be like static. It's just interphase, meaning that we've got chromatin there, not condensed chromosomes. So let's talk about prophase. Notice what's different here. Inside this region here, which once was the nucleus, we are starting to see the chromosomes condense. Here's a nice set over here. So one thing we're going to notice is the nuclear envelope breaks down. And the second thing we notice is these DNA molecules condense as chromosomes. And what that means when they condense, it means that they're starting to appear from the chromatin. So the chromatin is just like disorganized DNA hanging out in the nucleus. But chromosomes are organized in order for us to be able to separate that DNA into 2 separate cells. There's not much else going on in the rest of this cell, which I'm going to outline here. It is a plant cell once again, but in animal cells, that nucleus tends to have the same appearance. Notice in the picture on the right, you'll see early prophase, meaning that those chromosomes are just now starting to get condensed. And in late prophase, it looks like a swarm of snakes or worms inside what once was the nucleus. In fact, I don't see a nice looking nucleus in the picture on the right. Everything else here, these are all interphase. All of these are interphase. And I want you to compare how the chromatin appears in those nuclei, like here, compared to what you see here. Okay, so the interphase nucleus has much less organization, much more disorganization than the nucleus, or what once was the nucleus during prophase. This is metaphase in an onion root tip cell. Again, I know it's a plant cell because it's rather square, and sometimes people describe this as spiders dancing on a mirror. If I were to draw a line right through here, I would have what's called the metaphase plate. That's the middle region of the cell through which those mitotic spindle fibers will orient the chromosomes. Now, in human cells, we have about 46 total chromosomes. All 46 line up on this metaphase plate. There's quite a jumbled mess here, and there's a reason for that. If you have 46 chromosomes all lining up on that middle metaphase plate, it's going to look like a disorganized mess. But they're not separating, they're just being oriented here by those mitotic spindle fibers. This is a whitefish cell. It's an animal cell, not a plant cell. This cell has a more round shape to it. All of this stuff here that looks like a starburst, these are the mitotic spindle fibers oriented around a set of centrioles. Right here in the center, those are my chromosomes lined up on the metaphase plate. So here, the metaphase plate runs straight through the middle of this picture. Two more plant cells. Again, you have a square shape to those plant cells, and I'm looking for a set of chromosomes lined up dead center. Okay, and I can kind of see the mitotic spindle apparatus or fibers here. They're set up by centrioles. You can kind of see them. They're going to attach to that centromere and help to pull those chromosomes in too. In other words, during metaphase, those chromosomes are lined up like this along a metaphase plate. All right, so which picture here is metaphase? This one on the right is metaphase. Again, these are animal cells because they're somewhat round in appearance. You can see these chromosomes are bunched together in the center. The nucleus is gone. There's no nuclear envelope here. So they're disorganized looking, but really they're not. They're lined up on a middle plate that runs at an angle through there like that. In the picture on the right, we see, the picture on the left, we see anaphase over here. And this time we see chromosomes being pulled apart. In fact, these double-stranded chromosomes are being separated into two, okay? So we end up pulling double-stranded chromosomes apart into single-stranded chromosomes. That metaphase plate that was sitting right here through the middle, it's still there, but we no longer see chromosomes lined up in the middle. Now, anaphase means that this is the time when the sister chromatids, the arms of those chromosomes, are separated and pulled in two. This is a plant cell. It's rather square in appearance. And here I can see a set of chromosomes being separated from its neighbor. The metaphase plate was right through there, and those mitotic spindle fibers shorten and allow us to pull those chromosomes in two. So we end up having a set of single-stranded chromosomes in each pole or each end of the cell. Now, this may look kind of like metaphase, but look closely. You'll notice that there's a region of chromosomes here and here. But this cell looks like it's beginning to pinch in right through here. We're starting to see the formation of a cleavage furrow. That's what I'm going to look for to guarantee that I've got telophase. Now, these are chromosomes that will eventually be surrounded by a nuclear envelope once again. And I know that this is not metaphase because I'm starting to see this pinching effect because this is an animal cell dividing in two. This is a great picture for us to review all of the phases. First of all, let's start with a phase it's not part of mitosis. This is, of course, interphase. All of these pictures down here on the bottom, these are all interphase. And I know that because I see a distinct nucleus, I see scattered looking chromatin, and I see a couple of nucleolus or nucleoli inside those nuclei. Okay. Next thing I want to look for is prophase, and what I'm looking for is a condensed set of chromosomes in what was once the nucleus. This picture here on the far left looks best to be prophase. All right, so let's find metaphase. What am I looking for? Well, I'm looking for chromosomes lined up in the middle of the cell. That looks like a great picture of metaphase there. Notice those chromosomes lined up in the center portion. This one also looks like a great picture of metaphase. I'm again looking for those chromosomes lined up in the center. All right, and then this one here looks like late metaphase, early anaphase, because we're starting to see those chromosomes get pulled apart and separated. One more is this one here. This one looks like anaphase. I see the chromosomes being pulled in two, but I'm not seeing any kind of line form between the cells yet. Had there been a line there, I would have said that would have been telophase, but no line, I would say anaphase. This is actually what a cleavage furrow looks like. It's like putting a belt around a balloon and pulling till it pinches in two. So it may look like other things, but this is in fact a cleavage furrow that you're looking at. During telophase, we also experience cytokinesis, which means the separation of all of the other things besides chromosomes. That would include organelles in the cytoplasm. Let's take a look here. This one here looks like a great picture of interphase. Notice I can see a distinct nucleus. I can see chromatin, which is this disorganized looking DNA, and a nucleolus there. Okay, let's look for a prophase. This looks like a good prophase here. I'm starting to see maybe the nuclear envelope begin to break down in both of these, but I am seeing distinct chromosomes condense inside what was once that nucleus. Do you see a metaphase here? I think that's a good metaphase. You've got chromosomes lined up in the middle. I'm not sure I see a great anaphase, and I'll tell you what, this one here is probably early telophase. You know what I'm looking at? I'm looking at that line right there that's starting to form across the middle of this cell. That tells me this cell is finished anaphase and is probably into early telophase. In fact, these two down here look like they're in late telophase. That line just formed. See how those two nuclei seem to be fairly close to that line? That cell plate formed to separate those two cells. In fact, those two cells tend to be a little smaller than everybody else in this picture. That's what also makes me think that's probably late telophase. All right, so last set of looks here. At the top left, great picture of prophase. You'll notice it looks like a swarm of snakes in what is once the nucleus up here. That's prophase. Metaphase is our next one. That's this one here. That looks sometimes like spiders dancing on a mirror. And of course, those are all of the chromosomes lined up in the middle of that cell. Great picture of anaphase, top right. And I notice there's mitotic spindle fibers shown here in red, pulling apart those chromosomes into single-stranded chromosomes, which are pictured here in kind of a blue-purple color. And then finally, it looks like you have two newly formed cells below on the bottom right. That's telophase. And here you can see the nucleus is starting to reform around those colorful chromosomes. And you can kind of see where the cell is pinching in two here. All right, so one last look here. Late interphase. Notice there's starting to be some organization in the nucleus here, but by prophase, see how those two are different. Sit and compare interphase to prophase to guarantee yourself that you can tell the difference between the disorganization of interphase and the nice condensed organization of prophase. Metaphase has chromosomes lined up in the middle. Anaphase sees those chromosomes separate off the metaphase plate and get pulled toward opposite ends of the cell. And by the time we reach telophase, you start to see the reformation of a nuclear envelope around that DNA. You may also see a cleavage furrow if it's an animal cell, or a cell plate if it's a plant cell, dividing these in two. And by the time you reach the last part of telophase, you're actually seeing the opposite effects of prophase. The chromosomes relax back to their chromatin state, and then we see the nuclear envelope reform as the mitotic spindle apparatus disappears.